usually I'll say something like, it is my great pleasure to invite the speaker, but I am truly, deeply excited to invite the speaker today. So Danielle Nachman is our speaker, and she's a good colleague of mine. She's got a background in chemistry, but she worked on her PhD at Princeton University in the Geosciences department and graduated in 2021 when...
May 2020.
Yeah, when COVID was going on, and so we were just talking about how crazy that was. That was also the same time that I was transitioning from Princeton as a postdoc. But that's where we first met, and Danielle she went for a postdoc at Johns Hopkins Applied Physics Lab and became staff scientist there, and that's where she's at currently. And she does a lot of really cool research. A lot of the projects that they work on are relevant to the Department of Defense and things like that. So a great contact for any of you who are interested in these things. And today Danielle's going to talk about her research with Fenton chemistry, which has to do with iron and PFAS. And so I think it's going to be a great talk. So with that, Danielle.
Thanks, Jared. Thanks everyone for being here, and feel free to interrupt with questions at any point. So yeah, like Jared mentioned, I'm going to talk about using heterogeneous Fenton chemistry, which I will explain all of that as an approach for PFAS destruction, but also looking at other emerging contaminants that are relevant to human health, but also the Department of Defense. So I want to briefly touch on APL because I don't know if everyone here is super familiar with APL, although we get a lot of students and scientists that come from University of Maryland, but typically more from engineering side of things. But APL is a university affiliated research center. We're under the umbrella of Johns Hopkins, but we're very much our own entity. We're located out in Laurel, Maryland. And a lot of people know us mostly because of our work in space. So people are familiar with things like DART, the Double Asteroid Redirection Test, which happened last year, where we shot a satellite at an asteroid and changed its trajectory in space, which is way cooler than anything that I'm going to present on today. So, but we also actually do a lot of research in chemistry and biology, and we are expanding and growing our research in environment and climate as it becomes more and more relevant to the DoD. They're starting to recognize that climate change is a national security threat. So we have staff members with basically anything you could ever need help with. There's someone at the lab who does it, which is great. We have about 8,000 staff members. Again, we work with DoD, NASA, DHS, the intelligence community, so we have a very wide swath of sponsors and very interesting projects, many of which are at the classified level. And Ralph Summel, our director, likes to say our mission is critical contributions to critical challenges. So if anyone here is interested in APL, definitely feel free to reach out. So today I'm going to talk about PFAS and PFAS remediation solutions. I assume most people here are familiar with PFAS, but I will briefly mention sort of why we at APL are interested in PFAS, and that is because this is a major, major issue for the Department of Defense. The major contributor of PFAS contamination is actually from these firefighting foams, AFFF, aqueous film forming foams, that are used at DoD bases and facilities to put out fuel fires. And so you can see in the map here, there is a very large number of contaminated DoD sites. And so the government has become increasingly interested in PFAS remediation and this difficult problem because the water's contaminated, the soil's contaminated, and there's been a number of cases, like if you read about like Camp Lejeune, where people have been exposed to very high concentrations of PFAS. And it's estimated over 400 sites are contaminated, and remediation is going to cost DoD more than $2 billion. So they are very interested in effective solutions that are also low cost. I'll mention briefly just for, separation from water. Typically people use things like GAC and ion exchange resins. GAC is granular activated carbon. And today I'm going to talk about mostly destruction, which traditionally incineration is what's used, but that causes a lot of sort of downstream contamination. It's unclear what the byproducts of the incineration are. And so we really like to develop destruction processes that are more efficient, that produce non-hazardous byproducts. Yes, and I put there, of course, ensuring clean water has become a national security challenge as well. Today I mentioned I'm just going to talk about PFAS destruction, but I wanted to say that's not the only thing we do. We have a whole portfolio that's focused on PFAS remediation. So I'm happy to reach out if you're interested in any of these other things. We've developed novel membrane separation technologies for PFAS separation from water, and specifically from high concentration things like reverse osmosis, reject brine, and wastewater facilities. Fenton chemistry for PFAS destruction, which I'll talk about more. Part of the things that we're interested in is the byproducts removal. So in theory, when you destroy PFAS, you might end up with high concentrations of fluoride, which is something that we're interested in as well. Not just from a PFAS perspective, but also from a geogenic contamination perspective. So we've looked at methods for fluoride removal as well. And then this last piece is a project that I worked on for CERDUP, which is a joint funding agency between DoD, EPA, and the Department of Energy, where we are looking at replacements for PFAS in firefighting foams that basically can mimic the fire extinguishing properties without creating PFAS contamination. So I'm going to talk about Fenton-enabled water treatment processes. And so Fenton is not new. It's been around since, I think, the late 1800s in terms of, it's been around longer than that because it happens in the natural environment, but in terms of its study and sort of its understanding has been around since the late 1800s. And Fenton has been more recently looked at as a potential chemistry for wastewater treatment. But there's a lot of challenges to running Fenton at scale. But actually, this is a new wastewater treatment plant that's opening in Germany. It just went online in 2022, where they're actually using Fenton chemistry to destroy organic contaminants in wastewater. And typically what is done with Fenton chemistry is you use aqueous iron species, typically at low pH is the high efficiency, to create these reactive oxygen species that can go ahead and basically chew up your organic contaminants. And so people have looked a lot at homogeneous Fenton with these aqueous iron species, but the use of a heterogeneous catalyst for Fenton is a relatively newer concept, and the idea is that you can create much higher efficiency, potentially to destroy PFAS, which are much harder to degrade than many of these other organic contaminants. So, like I mentioned, people have looked a lot at Fenton chemistry with aqueous iron, but typically issues with that is it requires long UV exposure times. You need additional iron ions to help mediate the reaction. Sometimes you need to run it at high reaction temperature. So there's sort of all these barriers to especially running this at scale for wastewater treatment. So our idea was to look more into the heterogeneous Fenton, specifically using magnetite nanoparticles to improve the reaction efficiency. And so the idea is you have magnetite using nanoparticles, high surface area, efficient reaction, and that reacts with hydrogen peroxide in the presence of UV radiation, can produce reactive oxygen species and start to break down these PFAS. This is a very high level schematic. The magnetite nanoparticles in particular, so there's Fe2O3, it contains both, it's a multivalent iron species, it contains iron 2 and iron 3. So the idea also is that potentially this material is reusable because you're cycling between these oxidation states of iron. Some of the potential advantages of heterogeneous Fenton, you have, like I mentioned, recyclable catalysts, so you can, potentially, especially with iron, you can magnetically separate it from solution and then reuse the catalyst, which is not something you can do with aqueous iron. necessarily. You can potentially control the reaction if we can tailor specific iron-based catalysts to our organic contaminants of interest. Potentially lower environmental waste, easier to deal with, less harsh reaction conditions. We don't need to work at those really low acidic pH conditions, and potentially highly tailorable, which I'll talk a little bit about. So this is just to show you briefly what our reactions look like. Stick the PFAS in a beaker. At the very basic level, what we started with was just magnetite in the beaker, hydrogen peroxide, PFAS, see what happens, expose it to UV. We've actually transitioned from using this. This is a UVC microwave. We actually now just use a solar simulator for this. It's a video, but you can see you get this like very active evolution of oxygen and reactive oxygen species. I don't know if you can see it well here, but. All right, so the first thing we did with the magnetite was to look at destruction of PFOA and PFOS, which are two of the most common PFAS species. Those are the ones that have been most heavily regulated traditionally, and sort of the highest cause of concern, although now there are a few more that the government is regulating, the EPA, I should say. And we tested these at varied pH conditions and varied hydrogen peroxide concentrations. And also we did this 100 ppm of magnetite, and the bottom is with 1000 ppm of magnetite. Sorry, I know it's a little small, but on the left is PFOA, on the right is PFOS. And basically what we found is that, yes, if we amp up the pH up to pH 9, we get high reaction efficiency for degradation of PFOA and PFOS. But you can actually also get very high efficiency of destruction at neutral, like pH 7 conditions with the right amount of hydrogen peroxide, basically. So this is essentially to show that, again, benefit of using something like a magnetite is you can now run this reaction at a neutral pH, and you don't need to use those very harsh conditions that cause more downstream issues in the wastewater treatment process. I put here in the corner one thing just to note is that, and I'll talk about this more, but we wanted to show that this isn't just, I'll talk more about the destruction. This isn't just, absorbing onto magnetite, and that's why we're not detecting it in the system. You can see you actually have to have hydrogen peroxide and undergo the reaction to see this loss in PFAS species. So what I showed here, I should say, these are all measured with LCM SMS. We send this out to a lab that does the EPA method for detection of PFAS. And so that is a targeted analysis. So they go in and they just say how much PFOA or how much PFOS is here. The problem with that kind of analysis is that you're not getting sort of a mass balance. And if the carboxylic acid of the PFOA knocks off, then it won't come up as PFOA, but you still have this highly fluorinated compound there, right? And so how do we actually determine, is there destruction happening? And so for that, we use high resolution Orbitrap mass spec. So to look for these degradation byproducts. So you can see at the top, this is our control sample, the peak up there that's at 412, right there. That's the parent peak for PFOA. The second peak at 368, that's the decarboxylated PFOA that just happens from the ESI Orbi insertion. Now when we react it, so at low concentrations of hydrogen peroxide, and these were all done, I think these are at pH 9. Just for an example, you can see we're starting to reduce the signal of both the PFOA and the decarboxylated peak, and we're starting to see these other peaks come up. I'm not going to go into too much detail here, but basically these are different types of fluorinated compounds that are coming up. And then when we go to even higher concentrations of pH, we're actually completely losing that PFOA peak. And we're still getting some fluorinated compounds out, but they're sort of shifting more and more smaller. So what we see is this basically like subsequent cleavage of these CF2 groups off of the PFAS molecule. So we're getting destruction. The next step, what really needs to be done is mass balance with fluoride, which can be challenging because you have to adjust to the right concentration so that we can see that. We tested this method, so the top is pH 7, bottom is pH 9. We tested this against the 18 PFAS that are part of the EPA 537.1, so that's a standard measurement instruction or method for PFAS. These go, you can see across the bottom, they go from short chain PFAS all the way up to long chain. So the PFBS is 4-carbon chain. I think it goes out to about 11 carbon chain PFAS. And then the lighter gray bars are lower hydrogen peroxide concentration, and the darker gray bars are higher hydrogen peroxide concentration. So first of all, what you can see is pH 9, we're just getting better destruction in general. We can get 14 out of the 18 species, we're seeing, 80 to 90% destruction of both PFOA or of all of these. You can see the challenge here is these very short chain PFAS, PFBS, HF-POTA, which is Gen X, PFHXA. These are some of the ones that are starting to be regulated by the EPA. Those are much harder to destroy as you get shorter and shorter. Those carbon-fluorine bonds get stronger. So that's sort of, I think that's the challenge on everyone's mind of it works in the realm of PFAS destruction, which is you can destroy to a point and then you get, these short chains that are very recalcitrant and difficult to destroy. So that's sort of the next step of how do you tackle those short chains and, all these long chains becoming short chains, basically. I'll say, just a note, you can see that also at the higher concentrations of hydrogen peroxide, you actually get some reduced efficiency because you get this self-scavenging of the hydrogen peroxide. So you have to have this balance of basically ingredients in your reaction. So the other thing that we looked at is the recyclability of the magnetite. And so these were run, most of them through 3 cycles of PFOA and PFOS destruction. And then we analyzed for concentration of PFOA and PFOS. We found that there was basically no loss in the reaction efficiency, which is great. And you get sort of greater than 95% destruction of PFOA, even after 3 cycles of using the catalyst. So again, much more efficient to not have to prepare a new catalyst for every treatment cycle. One of the other things that we looked at with the recyclability is, do you get deformation of the magnetite structure, specifically the speciation and the oxidation state. I think I mentioned on this slide, so typically the iron 2 to iron 3, the 2-electron transfer is considered a stronger reaction. So one of the things that was of concern was if you continue doing the cycle, basically you oxidize all the magnetite and then it becomes ineffective. And so after each of these cycles, we collected sample and then used iron zanes. If anyone's familiar, this is a synchrotron-based technique. So this is iron cage zanes. I'm gonna jump over here. So these are our standards. The one at the top there is magnetite. These are some other iron species like hematite, iron foil, and wustite. And when we looked at the iron samples after different reaction conditions, multiple cycles, these all look like 100% magnetite. So we're not seeing any speciation change in the catalyst as well. So again, promising material for degradation. So I'm going to transition here a little bit. So basically we did all of this work. Okay, magnetite seems like it's a good catalyst. We're getting some destruction, and there's more work that needs to be done. But one of the things we want to understand, especially that question of how do you start to tackle like the short chain PFAS in particular, what we wanted to do was try to understand, if we can understand better what is the mechanism of destruction that is happening, maybe we can improve our catalysts, and tailor them towards this process. And so two of my team members, I brought on two team members with a computational background to do density functional theory, DFT calculations, to try and understand actually what happens to these PFAS molecules under Fenton conditions. And please don't ask me any questions about DFT because I cannot answer them. But I am not a computational scientist. But what we got was some really interesting results looking at this sequential oxidative degradation of PFAS. In some ways, this is kind of obvious, but you start to do the computation, you see that what we're looking at is a radical decomposition pathway, right? So these ROS, these reactive oxygen species interacting with the PFAS molecules. The calculations tell us that basically it's that specifically for PFOA, the decarboxylation, that first step is the rate limiting step. So once you can decarboxylate the molecule, then you can start to unzip and destroy the rest of the PFOA. What we see is when we model the non-oxidative degradation process, so basically just the PFOA in the solution versus in the UV Fenton conditions, that decarboxylation becomes spontaneous. So we're looking at the orange line versus the blue line. The blue line, it now becomes a spontaneous process to decarboxylate. Our question was, and we're still looking at, there's hydrogen peroxide in both of these systems. So our question was, does the PFOA coordinate somehow with the iron catalyst? And that is what leads to this decarboxylation and then the subsequent cleavage of the bonds. And so this is from some other papers where they've looked at the iron coordination with PFOA, and what our computational folks did was basically compute what would it look like on an IR if iron was coordinating with the PFOA molecule. And then we ran the experiment, and what you can see is this, the blue is just PFOA on its own in solution, and the black is magnetite in solution, the red is PFOA and magnetite. And when we add the PFOA with the magnetite, we're losing specifically this C double bond O from the carboxylic acid. And so we think that there is a relationship, the PFOA molecule is favorably interacting with the magnetite surface, decarboxylating, and then it can go on to react with the other reactive oxygen species, and then it's at the site of where these reaction oxygen species are being produced, so that can lead to the degradation as well. And so our question was, can we start to think about how would you modify this catalyst to more efficiently do this reaction, to start to degrade compounds better? And so we're starting, I'm going to just talk about this briefly because this is actually pretty new work. We are starting to look at other heterogeneous iron systems. So instead of magnetite, specifically looking at spinel ferrites. So these are magnetite-based materials that basically just have a transition metal substituted for one of the irons. The idea here is that you create an even more surface active site to interact with your contaminants. They're considered relatively low cost and you can recover them easily. And so our question was, can we, try this on PFAS? It's been tried for some other organic contaminants. You can see these are all there's a bunch of brand new papers looking at Fenton based on these ferrite systems. So specifically, we looked at cobalt ferrite for PFAS removal. So this is, you can see our system here. We have the solar simulator, the reaction. We pump air to mix the solution. And then we sent these out for analysis to look at PFOA removal in this case. And so what you can see is, so the two in the middle actually are just the magnetite on its own. And these are all done at pH 9. And different, so the top two are at room temperature, the bottom two are at 70 degrees Celsius. And so for the magnetite, we're getting about 60% removal at room temperature, 83% removal at the increased temperature conditions. But with the cobalt ferrite, under all the conditions, we're getting this very, very high removal. So it seems like the cobalt ferrite in particular is a good option. That was the first one we tried and it worked well. So we wanted to try some other transition elements as well. So we might still do that. But this seems to be a promising method. And then the sort of last piece I'll talk about here is, what about other emerging contaminants? So I mentioned we work a lot with the Department of Defense, and actually the group that I sit in particular, our main mission is detection and defeat of chemical warfare agents. And so there's a bunch of other stuff that we care about being able to destroy effectively, efficiently. And so we tested this reaction on glyphosate and acephate. So glyphosate is an organophosphate, acephate is like a pesticide. We also test these on simulants of chemical warfare agents, which I'm not presenting today, but it's very interesting as well. But basically what you can see is we're getting like complete destruction. These are in some sense easier to destroy than PFAS. So we're getting complete destruction of the glyphosate. We've monitored with phosphorus NMR and with this high resolution mass spec. So you can see that we have the glyphosate in solution. When we run the Fenton reaction, we're completely losing that, and we're creating these phosphate containing byproducts. And then this is similar for acephate, losing the parent peak. We're seeing cleavage of like the sulfur methane group on the acephate, and then getting these like phosphate groups coming off. One of the things that's interesting about this is that monitoring, especially when it comes to things like chemical agents, what actually comes off of that, sometimes they can be even more nasty than the first thing. So monitoring the degradation products is super important here. Yeah, so some next steps. Obviously, we want to continue improving on the mass balance. That's like a really important part of the PFAS analysis and looking at the reaction byproducts. We're continuing to work with our computational staff members to help understand the mechanism of the heterogeneous Fenton reaction, and keep looking at the ferrite system specifically with manganese. It might be an even stronger reaction that we can run at neutral pH conditions. And then we're going to start testing on a bunch of other emerging contaminants. Phthalates, microplastics is very big. Like I mentioned, CWAs and this 6-PPDQ, which is sort of an emerging contaminant. It comes from tires, and the DoD actually is very interested in it as well. So yeah, I'll pretty much end here. We're excited about this. We have a lot of stuff that we're thinking about. For example, one of the issues with Fenton is that you need to input so much hydrogen peroxide. So we're working on a system where we can produce hydrogen peroxide using carbon nitride on demand in the system instead of having to input hydrogen peroxide. And yeah, I think this has a lot of potential. Like I mentioned, highly tailorable, recyclable, and so I think this could be, implemented for wastewater treatment. And I think the hard part is how, when you start to translate this to scale, not just water treatment, also treatment of contaminated soils. You start thinking about air filtration, all this stuff. That's sort of the challenge here. That's where our systems engineers come in, start thinking about stuff. So, I'll just acknowledge quickly my team. They're all really awesome. They have great, like I mentioned, APL, basically anything you could want to do, someone knows how to do it. Our funding initially was internal funding from APL. We're now funded by the US Army, specifically DevCom Soldier Center. So they work on a lot of water contamination issues. Yeah.
Ask a question. It seems like from what I gathered, I am not a chemist, so you gotta forgive me if I come across ignorant about this
That's
question.
okay.
So what I gathered was, number one, if you have more saline type environment, like high pH, right? And you have some catalysts such as hydrogen peroxide, so you have more chances of basically breaking down, although the sort of getting that carbon breakdown is the initial step. So you will have more chances. Does that mean, let's say, if saline soils, they receive some of the sludge from these waste treatment plants that have not been treated for PFAS, that they have more chance of being remediated by high pH and maybe some catalysts such as hydrogen peroxide sprayed on it. I mean, this is a stretch, but...
No, it's not a stretch. It's something that, it sounds a little crazy, but it's something that we've thought about, right? Because you have these like vast swaths of contaminated soil. And so one of the, like in a high level theoretical way, wouldn't it be great if you could spray hydrogen peroxide and iron on the soil, it's exposed to sunlight and the things start to degrade, right? So that's kind of like the crazy thing we think about. And so the question is, how do you actually do that? I don't know if I have the answer, but one of the interesting things that we did look at, and I didn't present here, was we have looked at how does like saline environment and other ions in solution impact the efficiency of the reaction. We actually see that when you input a lot of like natural organic matter into the solution, it actually improves the efficiency because now you have more like basically more for the reactive oxygen species to react with and shoot off more reactive oxygen species. So that's pretty interesting. But then some of the stuff like very salty like can kill the reaction. Certain ions will clog up the catalyst, right? And so yeah, it's a lot of interesting questions that I don't necessarily have the answer to, but
because H2O2, it got my attention. I was watching this clip that it is actually free radical that in the body when oxygen, if H2O2 is in the body, actually free oxygen, one of the oxygen gets released and it damages the cells. That's where it can cause the cancer.
yeah.
So free radicals. So therefore they say, the types of vitamin C or other natural remedies that you eat, somehow they basically absorb those free radicals and they prevent from cell damage.
Yeah.
So I'm just thinking, connecting these
Sure.
together. Again, I'm not chemist, but this is a great work. Yeah.
Thank you.
Thank you for your presentation.
Thanks.
My question is related to how your team might, I guess, in the future, look at co-contaminants, because what you presented today were individual PFAS compounds.
Yeah.
And so with sites available on your catalyst, how might that work if you have more than one contaminant present in that system?
Yeah, absolutely. It's something that it's actually part of the work that we're doing now. but it's like, you know, the third task for the sponsor, so we haven't gotten to it yet. But yeah, that's it's a really good question, right? Because some of these things, like I showed, like acephate and organophosphates, glyphosate, right? Like they react super readily. So if they take up all the sites on the catalyst, then maybe we're not getting to the PFAS, right? And so that's something that we need to look at specifically about like the kinetics and the thermodynamics there. I think the other piece is, you know, again, depends on where you want to use this, right? Sometimes, especially with PFAS, one of the things that they look at is, you know, after they've done all these other treatment processes that have basically, right, like GAC is going to pull out most of the organics, organic contaminants, and then, you know, you end up with this like reverse osmosis reject brine that's like gross and has a lot of salt. PFAS is one of the major contaminants because it's just not getting caught by anything else, right? And so I think this can still be useful for PFAS depending on what you're trying to treat. But yeah, the co-contaminant issue is super important.
This is a great talk, thank you. But I was just wondering, and again, forgive me if I'm using the wrong words, but for the analysis that you did using the cobalt and the iron, are there differences with the packing structures of those, like magnetite versus that like play? Like, why did that react so much?
My understanding is that I mean, yeah, you're gonna get like a different, like a deformed basically structure with the transition metal in there, and so yeah, I think it's just the transition metal is like a really good bonding site to bring in the contaminant, but yeah, I don't know all the details off the top of my head right now. I'm sorry. Yeah.
Yeah, off of that question, so like, I mean, yeah, having the different, basically like doping the iron minerals with different elements can make it more reactive or change the reactivity. I mean, naturally occurring iron minerals have all kinds of like extra stuff.
Yeah,
associated with
exactly.
them too. And so I guess like on that, another way to alter the reactivity of the magnetite would be to play around with its crystallinity, like nanoparticles versus like larger crystal structures.
Yeah.
Have you guys tried?
No, so that's actually something that Hunter in particular, I think he's like a catalysis expert, and he is super interested in that kind of stuff. To date, we've basically just been we just order like these catalysts. But they're also super easy to. They're cheap, but they're also super easy to synthesize. And so, yeah, one of the things we've thought about is if we synthesize them ourselves, can we? You can actually even order like different nanoparticle sizes and stuff, and yeah, test how well they do.
Yeah, and that was really just a segue into like this big thing that I wanted to throw out. Because like at the end when you're talking about, you know, thinking about engineering solutions, and maybe this kind of goes back to what Adele was talking about too. And I might be able to drag Andy into this maybe. So the first thing that came to my mind was a wetland, like a constructed wetland. Because I think the wetland plants probably excrete hydrogen peroxide. I don't know if they would only do it because they're stressed or if they would do it like in a regular way.
Yeah.
So that would be a natural source of hydrogen peroxide. And if you just like dumped in iron oxides like goethite, like iron oxyhydroxides, there's iron producing bacteria there that could transform it into magnetite. Yeah. And the beauty of that is you showed in your work, which was cool, that the catalyst is so efficient, the iron to iron 3 ratio pretty much holds. But in the natural environment, if you were worried about the catalyst being completely oxidized, if the microbes are there to reduce the iron 3 back to iron 2, that could be a benefit.
Yeah, for sure. Yeah, if you think about it's sort of getting back to that question of like how would you remediate like a contaminated soil?
Right.
You know, if you could inoculate it with some sort of microbe. I'm not a microbiologist, but you know, inoculate it with something that helps sort of keep the redox balance.
Yeah.
That'll be interesting.
How will this work, like, meeting the UV if this is something like not on the surface level?
Sorry, say that again?
Like if you need the UV light to continue
Yeah. That's a good question that I don't have the solution to yet, but, no, it's like, I think that's the other problem with like, so in like wastewater treatment facilities, right, they have these like massive tanks that have UV exposure, so that's not a problem. But yes, like if you wanted to implement this in soil, right, you would, just be very much at the surface, for the light penetration, so that's definitely an issue. And so, yeah, I think so Hunter was working on this idea of like producing the hydrogen peroxide on demand so that you don't need to continuously run the UV also in the system. And so maybe that's a solution, for implementing in the soil system. Yeah.
You may not know the answer to this, but
Yeah.
it seems if in Germany they have built a $300 million wastewater treatment plant that's using this technology.
Yeah.
I don't know what my question is. Are they targeting a whole host of different contaminants, PCBs and other things? Are they getting,
Yeah.
how are they applying that technology?
Yeah. So it's interesting. So, because I was looking into this a little bit. There are other Fenton plants, but they're typically for like industrial chemistry, like cleanup kind of stuff. This is the first one that's specifically for wastewater treatment. I don't know off the top of my head, like what specifically they are targeting. I do know that they're doing like aqueous homogeneous Fenton, at this stage, so.
There's a question from someone on Zoom. So Danielle Sanchez actually works with Dr. Duncan.
Mhm.
His master's work is on looking at PFAS contamination like around Fort Meade area.
Mhm.
So this question, it says, hi Danielle, thank you for a great presentation. I know that the EPA put out a recent plan to regulate several PFAS in drinking water, though from what I understand, PFAS treatment technology still has a lot of research gaps to fill to meet the needs of drinking water plants. I was curious to know if that regulation decision has affected your research at all, perhaps in the pace of the research or the attention that other organizations have given.
Yeah, for sure. I will say a few things about that. First of all, it's interesting if you look at the regulations for the PFAS contamination, they're set at what their instruments can measure, which is very interesting. So probably the contamination levels that they're concerned about are even lower than what the limits are. I think effectively zero is what it should be. But yes, because of that, now that they're regulating those 6 PFAS and it's at the federal level, and so now that's become sort of like a very big issue. Like I know the White House put out like a strategy on PFAS last year. And so there's a lot of attention on this. So, yeah.
May I ask another quick question?
Yeah.
So, waste treatment plants, so there's the sludge part of it and there's the water part. The reason I'm asking this, both in California, many parts of the world, and including us, we are trying to see how we can use that treated wastewater to tertiary level for irrigating non-food crops. So the question is that, are the PFAS more actually are in adsorbed form, just like some pesticides are, for example, particulate form, or are they more in aqueous solution form that treated wastewater, if they haven't treated in waste treatment plant for PFAS, then that water actually can have PFAS, which can then distribute into their soil system by irrigation and so on. That's the question.
Yeah, I would say it's in both, and I think that's sort of the big challenge with PFAS is that PFAS is not just a one molecule, and it's, the EPA, now the EPA 1633 is looking at 40 different PFAS, but there's actually tens of thousands of different types of PFAS, right? It's anything that's fluorinated, and so, the long chain PFAS are gonna absorb into like the sludge and the biosolids and things like that. The short chain PFAS are, much easier to get into the wastewater and all of that. So it's a big problem for everything.
We have a couple more minutes if there are any other questions.
So Danielle, for your talk, thanks.
don't know why, I was just very curious about why this reaction is called heterogeneous Fenton reaction. I'm wondering either like a homogeneous Fenton reaction also exists.
Yeah, it's just a differentiation basically. Like homogeneous Fenton is just aqueous iron species, so the reaction happens in the solution, versus heterogeneous is you have some solid catalyst, so the reaction is happening on the catalyst site. Yeah.
So anyone
It's a
who's taken
fancy
my
way.
general soil chemistry class has received that question.
Yes.
Yeah.
How many people got it right? Sorry.
I'm still trying to figure out if the answer to it or if it was ChatGPT. Yeah, so actually, I guess just one more question I had, and in this, so I feel like I've read somewhere that with the heterogeneous Fenton reactions, and again, this might have to do with the crystallinity of the mineral, that it might be possible to generate free radicals even without hydrogen peroxide.
Yeah.
Do you know anything about that, or?
Yeah.
Yeah.
Like just from the catalyst itself.
Yeah. Like, and it might be because like you have to have like some like you said, natural organics.
Yeah.
That helps. You
Yeah, I think you would need some sort of something that can shoot off the radicals
Right,
in the first place.
Right.
So if you were just doing it in like DI water, I don't think you would get any